My name is Laura Snow, and my partner and I are Shia Carr, our research assistants in the Riley Lab at the University of Massachusetts Amherst. Today, we would like to discuss our examinations of the potential of Nisin as a treatment for bacterial speck of tomato.
Bacterial speck of tomato is a plant disease that, in fact, you guessed it, tomatoes. It's caused by the bacteria Pseudomonas syringae, which is one of the most common plant pathogens, with over 60 different infectious strains alone. And these strains have an extremely large host range of over 180 different plants. I'd argue that tomatoes are one of the most valuable. In 2020, they had the highest total market value of all vegetables that year in the US alone, valued at $1 billion. And they're grown a lot as well. In 2014, 171 million tons of tomatoes were produced. The symptoms of bacterial speck present as these small black and brown spots, sometimes with yellow halos, that you can see in the images on the right on the plant tissue. This disease can stunt plant growth and delay fruit maturity, and in extreme cases result in complete crop loss. This disease can devastate entire fields of tomatoes, and so that makes it extremely important to find ways to prevent and treat this disease. The current treatment methods of bacterial speck revolve around prevention. The first thing that farmers try to do is prevent crop loss from ever occurring in the first place. They do this by first removing the debris from diseased tissue and destroying infected plants rather than trying to cure them, because once disease symptoms begin to show, it's extremely difficult to treat bacterial speck. They also have to rotate fields as the bacteria can overwinter in the soil and infect a new season of tomato plants, even years in the future. And a switch to drip irrigation systems also helps prevent infection. This is because conventional sprinklers can splash the bacteria to plant tissue, and drip irrigation systems circumvent that. Chemical treatments are also in use. Copper-based sprays have been used to prevent bacterial speck since 1885. However, they're becoming increasingly ineffective as cases of copper resistance have begun to be shown since the 1980s. Copper is also toxic to the plant tissues and can leave residues on the fruit. The other alternative are antibiotics, primarily streptomycin, which has been in use since the 1980s. However, just like any other antibiotic, there are many resistant strains of Pseudomonas syringae, which makes this drug also ineffective. So to summarize, the downfalls of current treatments are that they're toxic to plant health, they're ineffective at clearing infection, and the resistance to these treatments is rising.
Bacteriocins are proteins that are naturally produced and secreted by bacteria of all different kinds. They have antimicrobial properties, which means they can kill other bacteria. These properties often act on a narrow spectrum, which means they are specific to the bacteria they are killing, which are often closely related to the producer bacteria. Bacteriocins are generally non-toxic and environmentally friendly, which is important as many conventional antibiotic drugs are not as environmentally friendly. Nisin, the bacteriocin of our study, is approved by the FDA as a food preservative, so it is safe for human and animal consumption, and it is the only bacteriocin currently approved by the FDA. It has killing abilities against gram-positive bacteria, which have been previously shown in literature. However, Pseudomonas syringae is a gram-negative bacteria. Bacteria are predicted to have a low resistance to Nisin, as this resistance can only be induced to be involved in a laboratory setting and has not yet been observed in an environmental setting. This is especially important as many bacteria species have evolved very high resistance to conventional antibiotic drugs, making them much less effective. The purpose of our study is to determine if Nisin, the bacteriocin, is an effective treatment for bacterial speck of tomato, which is caused by Pseudomonas syringae. We will evaluate Nisin's efficiency through minimum inhibitory concentration tests, also known as MICs. First, to test the efficacy of Nisin against Pseudomonas syringae, we completed an assay to see if Nisin at the highest concentration available to us, which was 4.23 milligrams per milliliter, had the ability to kill Pseudomonas syringae bacteria. We found that all of our strains could be killed by Nisin. We also tested the solution Nisin was suspended in, called citric acid buffer, to determine if the bacteria was killed by the solution altogether or by Nisin itself. The image of the agar plates on the left show a sample of a few of our strains tested with spots of Nisin in citric acid buffer on top displaying clear inhibition and bacterial killing, while solely citric acid buffer spotted on the bottom did not kill bacteria. Overall, Nisin had a greater killing ability of all strains tested than the suspended solution. In order to determine the minimum inhibitory concentrations of Nisin against each strain, we grew a bacterial culture overnight and then prepared a 0.5 McFarland standard of 1.5 times 10 to the 8th cells or colony-forming units per milliliter. We then prepared a 96-well plate with varying amounts of lurea broth, the bacterial growth medium, cells diluted to 10 to the 5th colony-forming units per milliliter, and varying concentrations of Nisin. We tested neat Nisin, which is 423 micrograms per milliliter, all the way to the 7th dilution of Nisin, which is only 3.3 micrograms per milliliter. After letting the bacteria grow for 24 hours in an incubator at 30 degrees Celsius, we scored the plates visually, determining if there was bacterial growth or not in each well. The chart on the left is the template we used to fill the 96-well plate. The figure on the top right displays the general contents scaled in a single well.
Here are the current results of our minimum inhibitory concentration study so far. We have generated MICs against 15 strains of P. syringae so far. This is by no means enough strains to suggest an MIC 50 or an MIC 90, but can give us an idea of where future MICs might land. The range of our MIC so far is between 6.6 micrograms per milliliter to 52.8 micrograms per milliliter, with the majority of our strains having an MIC of 13.2 micrograms per milliliter. This is an extremely low concentration of drug and is less than or comparable to the current chemical treatments that we're seeing in the literature so far. At the current stage of our project, so far, we are able to conclude that Nisin is effective at inhibiting the growth of P. syringae in a laboratory setting. It's inhibited the growth of all strains that we've tested it against, and the MICs that we've generated so far have been extremely low. So only a very small amount of Nisin is required to inhibit the growth of 10 to the 5th CFUs. We have several studies in progress right now, including a continuation of our Nisin MIC study with more P. syringae strains that we've been lucky to receive from the Martin lab at Cornell. This will help bolster our current MIC data to give us an even better idea of where the MIC of Nisin is against P. syringae. In parallel, we're conducting streptomycin MIC studies to directly compare against Nisin MICs. This will help us directly compare if Nisin or streptomycin is better. And finally, we are determining the spontaneous resistance mutation rate of P. syringae when treated with Nisin. And eventually we will generate it against streptomycin. The goal of this study is to determine if P. syringae has less of a chance of becoming resistant to Nisin as compared to streptomycin. This will help us, again, make a case that Nisin might be a better treatment for P. syringae. Some future directions for this project are determining the best method of drug application, and of course, testing the effectiveness of Nisin on tomato plants in the field. Thank you to our PI, Dr. Margaret Riley, and our other team members, and of course, the Martin lab, who have given us some strains. Thank you very much.